by analyzing large-scale genomic features. That is, we cannot work with the genome at the nucleotide level. Instead, we define markers that are short sequences, highly conserved sequences over the genome that are still long enough such that their conservation is statistically significant. So once that complete genomes are rapidly becoming available, we wish to compare these genomes at once. Therefore, working at the marker level it's not possible anymore, as it would produce computational bottlenecks. Therefore, we use the markers to identify and define large-scale patterns, and this is known today as Synteny blocks. If we use Synteny blocks systematically, then basically we can obtain an alternate representation of the genome in terms of such units. So, what are the benefits to using Synteny blocks for large-scale comparative studies? Well, as they define a next level, a higher level of abstraction over the markers, they reduce automatically the data, therefore they reduce the computational complexity. In the same way, they provide the filtering, they filter out basically the variability among individuals and reduce the dependence on evolutionary models that would be specific to certain genomic regions only. And as we define Synteny blocks, we wish to define significant units such that they are used in targeted studies, so this is how they facilitate such studies. So what do we mean actually by Synteny blocks? Let me illustrate it on a pair of genomes. When we parse the genomes and compare them against each other, we wish to identify blocks, so large chunks of DNA that are highly conserved, and such that afterwards we can match them together and form Synteny blocks. Here we see, for instance, two pairs of Synteny blocks in yellow and green. And what we expect to see inside a pair of Synteny blocks, well, it's a set of shared markers. As we can see, the markers are related to each other, that's indicated by the vertical lines, and the markers need not be like unique copies. As we can see in the second block, the pink marker appears in a double copy, or at the same time they can be singletons, just like the rightmost marker in the first block. So such a set of markers, such two blocks, define a pair of Synteny blocks. The concept was introduced first in 1971 by Renwick on a study on the human genome, and it stands for the collocation of markers on the same chromosome. So later in 1984, Taylor and Taylor, they introduce, they extend the notion to a set of markers that is conserved between two genomes, where the markers conserved, the ordering and the orientation of the markers between the two genomes is conserved. So this is what loosely defines co-linearity. And as we can quickly see, such notion does not scale to the comparison of distantly related genomes. Therefore, in order to address this shortcoming, later studies have relaxed this notion. So we speak about a relaxed co-linearity. That is, in this notion, the markers, the set of markers shared between two genomes need not be in the same ordering and orientation. However, this still stays at the level of pairwise comparison. So then later on, when multiple genome comparison became possible, Synteny mining tools started being developed based on heuristics. So this means that there was no a priori definition given for the structure that would indicate the structure of the resulting Synteny blocks. That is, we cannot quantify such blocks and we cannot compare them against each other. And most importantly, we cannot improve such tools. We cannot evaluate them. So in order to address this issue, we propose a formal definition that we build it on homology, because it is a general and flexible enough notion which requires only common ancestry. In the same time, homology is also a transitive, presents the transitive property that allows the definition of equivalence classes. That is, we can define families of homologous markers and genes, and if we define synteny in terms of homology, it will automatically benefit of the properties of homology. So in the same vein, we can define families of homologous synthetic blocks. Now this notion I will be using quite often throughout this presentation. So let me present you our definition. There are two main requirements and an optional one. So we call two or more blocks to be homologous synthetic blocks if and only if the blocks are connected through homology statements between their markers, and the markers which are part of such homologous synthetic blocks are related to other markers within these blocks and not only to markers outside these blocks in unrelated blocks. Now, if we wish to emphasize conservation, then we can also require that the markers form a frame. So what is the frame? If the two end markers of the synthetic blocks are homologous counterparts for each other, then they define the frame. And if we have such a frame, then we basically set very well defined boundaries on the range of these positions, the positions that form a synthetic block. So this is a lot of theory. Let me please introduce the notion better on some cartoons. Let's illustrate the first requirement of the definition. So what we define as synthetic blocks are a set of markers within the same genome. And then a synthetic block family is a set of synthetic blocks defined throughout several genomes. The second requirement of the definition is related to the homology between the markers of the synthetic blocks that are related, and this is best illustrated through how the property is violated. So here we have the pair of red markers, and as we can see, the red marker in the first genome in the leftmost synthetic block family has its only counterpart, which is its only homologous counterpart. It's situated in the middle synthetic block family, and it doesn't present any homologous counterpart in its own synthetic block family. So this is a violation of the second requirement of our definition. Now, the framing property, and it's illustrated as we can see on the left synthetic block family. Here the frame is very neatly defined by families of homologous markers which are situated on the same side of the synthetic block family. But in the same time, the example on the right side, it's just as valid as the one on the left. It's just that the framing marker families belong to two different sides, and they are treated by the blue and the green color. So, given the formal framework that we have defined, we choose to evaluate three state-of-the-art tools that are capable of comparison of multiple genomes. And we evaluate them on a data set of 8 these genomes that, as we can see, they present high variability at the genomic structure level. They are quite distantly related, but we have also quite closer related structures such that we obtain an optimal analysis. So then the output of the tools on this dataset is going to be synthetic block families, and this is what we are going to work with. So given the framework, given the experimental setup, we define measures. So the first measure is for the violation property, the second property of the definition. The second measure is going to quantify synthetic block families based on the percentage of its markers that relate to at least another marker in the synthetic block family. The third measure takes the second one a step further and actually requires that each marker within a synthetic block has at least one homologous counterpart within every other synteny block part of the same synteny block family. As we will see, for the three measures, the tools will exhibit, well, the results produced by the tools will exhibit different behaviors. So we will present also a fourth measure, which will actually show the commonalities shared by the three tools. So let's start with the first measure. This is the case that we are evaluating. And as we can see on the plot on the right, well, Syntenator and Iador produce synteny block families that contain very few to no markers which don't relate at all to the other markers which are part of the same synteny block family. And whereas Dream Synteny actually produces synteny block families which contain markers that are very sparsely related to each other. The second measure is requiring that each marker within a synteny block part of a synteny block family has at least one homologous counterpart in the whole synteny block family. So as we see on the plot, Iador and Syntenator produce synteny block families which obey this rule quite well, while Dream Synteny follows the opposite trend. Now, if we take this measure further, and we require that each marker has a homologous counterpart in every other synteny block, part of the same synteny block family, then we see that there is a huge change in trend in the synteny block families produced by Iador. So here the tool is less picky about the markers that it places in a synteny block family. And it leaves only Syntenator to be producing synteny block families which are densely interconnected through the required homology statements. So these results were showing different behaviors of the tools. So let me illustrate the commonalities. So for this illustration, out of the synteny block families produced by each tool, we chose only the synteny blocks of the Candida genome, and each slice of the ring represents a concatenation of the synteny blocks produced by the tool in the same order that the blocks appear on the genome. So, and the fact that for Iador I split the slice is just for visualization. So as we can see, there are a lot of thin bands which represent basically the correspondence between the blocks that they link together. So as we see here, for instance, Iador's, the small blocks produced by Iador from this position, they would relate to some blocks produced by Syntenator at the corresponding position. However, what is important to see here, it's depicted by the dark blue band on the blocks, well, block concatenation produced by Iador, where we see a certain genomic region which is covered by huge chunks of, by large blocks that are very similar in content, and they are produced of a similar manner by the three tools. So this shows that the tools are able to identify and define similar blocks as well. So what we have seen so far is that the tools exhibit very different behaviors and produce different structures. But we see that to some extent they also share common structures, so they have common features. But at the end of the day, what we want is to find like a common denominator, a normalization between these two behaviors, and this can be achieved by mining for synteny in a principled way. So, and we don't want at the end of the day that all the tools produce the same structures, because this would basically not benefit targeted studies, which from the use of synteny blocks. So then, in conclusion, the problems that we have addressed and the solutions that we have provided. So we have seen the evolution of the notion of synteny. We have seen that there is no commonly accepted definition for Synteny, for syntenic blocks. Therefore, we propose a homology-based principle definition that later on we use to actually evaluate and compare syntenic blocks. Therefore, we provide, besides the generic genome coverage-based evaluation, we provide extra insight into the structure and the composition of syntenic blocks. And in this manner, we basically address the shortcomings of the current tools, and we provide an integrated solution to develop better heuristics and tools. With this, I would like to acknowledge my supervisor, Professor Murray, for his priceless support and insights, and to the members of the lab for the very valuable discussions. I would like to acknowledge the NSF for the funding provided for my travel to ISMB 2014. Thank you very much for your attention, and I'm very happy to answer your questions.
So thank you very much, Cristina, and particularly for enduring such difficult circumstances. You've got us back on track time-wise. So do we have any questions for Cristina? Okay, while you're thinking, okay, Paul.
Yeah, thank you. That was really interesting. I think it's a worthwhile endeavor to try to identify. We really come up with a definition of what we mean by synteny. So to me, this homologous marker-based approach,
yes,
it looks like it would be amenable to using alignment methods and generalizations of alignment methods, so alignments of homologous markers to start defining blocks. Do you think that's an approach you would consider in the future, or how do you imagine turning this into algorithms for analysis of synteny?
So, just to be sure, is your question about how to identify homologous markers?
So how, given a set of homologous markers,
yes,
right, or how would you actually go about constructing larger blocks from them?
Well, besides the fact that the definition is very idealistic, there exist also other notions that I have not presented here, like, for instance, common intervals. So this gives also a very good structuring on which we could build implementing, and then that notion we could use to expand it and adapt it to the data set, and then this way we would define, for instance, we would identify, try to identify synteny blocks. So
by intervals you mean distances, like genomic coordinates?
Well, not necessarily the coordinate, the precise coordinate, but the structure that is behind it. It's this identifying the set of markers, and but allowing in the meantime some rearrangement in the structure. So the concept is very well mathematically already very well defined and set, and it presents quite a few features which match very well the definition, which is very idealistic. Right. So then marrying the two together, that would be, for instance, a way that we would think of pursuing.
Thanks. That was great.
I hope that answers. Thank you.